All right, welcome. Today, we're going to pull back the curtain on this amazing scientific tool, an unseen detective, really, that keeps our world safe every single day. But to really get a grip on its power, we've got to start with a question that sounds, well, pretty much impossible.
I mean, seriously, how would you even start? It sounds completely impossible, right? Kind of like trying to find one particular grain of sand on a massive beach. But here's the crazy part. There's a technology that can do something very, very close to that, and with just jaw-dropping precision. So let's see how it works.
Okay, so our solution starts with this machine. Think of it as a super sophisticated sorting system, but for molecules. Let's meet the first half of our detective duo, gas chromatography.
And yeah, that racetrack analogy, it's perfect. Imagine you've got this jumbled up crowd of all these different molecules. Well, GC's job is to make them all run a race. It separates the super fast ones from the slower ones until every single one crosses the finish line all by itself.
So how does this race actually happen? Well, it's basically a three-step process. First up, you inject the sample mixture. An oven heats it up like instantly, turning it into a gas. Then a carrier gas, which is kind of like a powerful gust of wind, pushes everything into this incredibly long, super thin, coiled up tube they call a column. And inside that column, that's where the real magic happens. The lighter molecules, they just zip right on through. But the heavier, stickier ones, they lag way behind. And by the end, they all pop out of the column one by one, perfectly separated.
You know, even the wind they use to push everything along, that carrier gas, that's a big decision. Helium is a super common choice because it's really efficient and it's totally inert. That means it won't mess with the sample at all. The downside, it's a finite resource and it can get pretty expensive. So the other option is hydrogen. It's way faster, which can speed up the whole analysis, but it's also flammable. So right from the get-go, scientists are playing this balancing act between speed, cost, and safety.
Okay, so we've sorted all the molecules. Awesome. But we have no idea what they are yet. That's where the second half of our detective team swoops in, mass spectrometry. This is the expert identifier.
Yeah, if GC is the one sorting the suspects out of the crowd, mass spectrometry is the one that pulls each one into the interrogation room. It takes every single molecule that crosses that finish line and figures out its exact identity, basically by weighing it and then smashing it to pieces.
So how does it create this fingerprint? It's this really cool four-step process. First, the molecule gets zapped with a bunch of energy, which gives it an electrical charge. But that jolt of energy also shatters it into smaller, predictable pieces. Then, the machine analyzes all those little fragments, measuring the precise mass of every single one. And the pattern that comes out, it's totally unique. It's a perfect, unmistakable molecular fingerprint for that one specific molecule. No two are alike.
This analogy just nails it. It really does. Imagine there's a car crash. A really good investigator could walk up to the wreckage, look at the engine block, a piece of the door, the shape of a taillight, and they could tell you the car's exact make and model. That's exactly what mass spectrometry does, just with molecules.
Okay, so we've got the master sorter and the master identifier. What happens when you put them together on the same team? Well, you get an absolutely unbeatable detective duo.
And that's GCMS. It's brilliant. The gas chromatograph does its thing, carefully separating this complex mess, and then it sends each pure chemical, one after the other, straight into the mass spectrometer. The MS then grabs each one, shatters it, gets that unique fingerprint, and boom, positive ID.
But wait, how does it know which fingerprint belongs to which molecule? I mean, it's just a pattern, right? Well, it uses this massive digital library, like the one from NIST, the National Institute of Standards and Technology. Think of it like a police database with millions and millions of known fingerprints. The machine just searches for a match, and the second it finds one, our unknown molecule is identified. Case closed.
All right, let's circle all the way back to where we started, that drop of poison in the river. Let's see how our detective duo, GCMS, solves a real-world contamination case, step by step.
So here's the setup. We've got a river flowing near an industrial zone, and there's a real concern it might be contaminated with this nasty pesticide called chlorpyrifos. So a scientist heads out and takes a water sample to find out what's really going on.
Okay, so before they can even test the river water, the scientist has to teach the machine what it's looking for. It's like training a dog. They run several samples that have known amounts of chlorpyrifos in them. This creates what's called a calibration curve. And if you look at the data, you can see it's super clear. The more pesticide there is, the stronger the machine's response. It creates this perfect, predictable, straight-line relationship. This is the ruler they're going to use.
And now, it's the moment of truth. The scientist runs the actual river water sample through the GCMS. It does its separation thing, the analysis happens, and the detector spits out a single number, 18,500. Okay, so what in the world does that number actually mean?
Well, remember that calibration curve we just made, a ruler? Using that, the scientists can translate that raw number, 18,500, into a real-world concentration. And it turns out, a response of 18,500 means there are exactly 1.85 milligrams of that pesticide in every liter of water. We finally have our number. But the big question is still hanging out there. Is that a little or is that a lot?
To figure that out, we've got to have some context. We need to compare it to the official safety limit. And according to the EPA, the absolute maximum amount of chlorpyrifos that's considered safe in drinking water is just 0.3 milligrams per liter.
And there you have it. The verdict is in. The river water isn't just a little contaminated. Its contamination level is over 6 times the accepted safety limit. Our unseen detective just uncovered a major environmental threat, and now the authorities know they need to take urgent action.
And look, this is just one example, right? The truth is this incredible technology is working all around us, every single day, in ways you might never ever expect, all to keep us safe.
Just think about your day so far. Did you eat a strawberry for breakfast? Well, GCMS probably checked it for pesticides. What about the air you're breathing or the water you drink? Yep, constantly being monitored with this technique. It's absolutely crucial in forensic science for identifying drugs or poisons at a crime scene, and it's used in hospitals to make sure patients are getting the perfect dose of their medicine. It really is an invisible guardian.
So, we went from a question that sounded completely impossible to a tool that protects our food, our environment, and our health. GCMS is just this powerful reminder that sometimes the most important work is being done by detectives we can't even see. It really makes you wonder, doesn't it? What other invisible guardians are out there, quietly keeping our world safe?